Welcome, everyone. I want to begin by acknowledging that we are gathered here at Carleton University on the traditional and unceded territory of the Anishinaabe Algonquin Nation. We recognize the Indigenous peoples' long-standing presence in this territory, and this recognition and respect for Indigenous peoples and their territories is a key step towards reconciliation. So welcome, everyone. My name is Maria DeRosa, and I'm the Dean of Science here at Carleton University. On behalf of the Faculty of Science, I would like to welcome you all to this event, our 2022 Herzberg Lecture. The Herzberg Lecture is hosted annually in honor of Dr. Gerhard Herzberg, who taught and conducted research at the University of Saskatchewan and the University of Chicago before moving to the National Research Council in Ottawa in 1948. He is widely recognized for his work in the field of atomic and molecular spectroscopy, and in 1971 was awarded the Nobel Prize in Chemistry. In 1973, Dr. Herzberg served as Carleton's chancellor, did so for 7 years, leaving a lasting legacy that includes being the namesake of the Faculty of Science, one of the Faculty of Science's buildings. Throughout his career, Dr. Herzberg was dedicated to furthering freedom of inquiry in science and a strong proponent of the important and relevant role of science in society. A lecture series in his name, exploring a science-related topic of general interest, seems a fitting tribute for his research and work. This year, our lecture will be delivered by Dr. Sarah Sally Otto. Dr. Otto is a Killam University Professor at the University of British Columbia. Dr. Otto is known for theoretical studies investigating how biological systems evolve, with over 200 publications and a book. Was a co-founder of the Canadian Society of Ecology and Evolution, and she has recently served on the Species at Risk Advisory Council to the Minister of Environment and Climate Change Canada. Dr. Otto also launched and directs the Liber Ero program, a Canada-wide postdoctoral program in conservation science. And during the pandemic was involved as a co-leader of the BC COVID-19 modeling group and the CoVaRR-Net computational biology and modeling pillar. Dr. Otto's awards include a MacArthur Fellowship, a Steacie Fellowship, a Steacie Prize, and a fellowship in the Royal Society of Canada, the American Academy of Arts and Sciences, and the National Academy of Sciences. It is now my distinct pleasure to welcome Dr. Otto to present the talk, Evolution in a Rapidly Changing World. Dr. Otto, the floor is yours whenever you're ready.
Thank you. My pleasure to be here, at least virtually, and join you and share thoughts on evolution in this rapidly changing world. I'm going to share my screen and hopefully all will be well. Just go into full view. Do I have you with me? Can you see the screen? Awesome. All right. And I would like to start with acknowledging that I am calling in today from the traditional and unceded territories of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations. You know, if you're like me, when we were children, I remember driving along the road and rolling down the window. And when we were done with our snack or our lunch, we'd throw out the wrappers. And I remember asking my dad when I was a young kid, what happens to all those wrappers? And, you know, I think his response was, well, you know, eventually they decay and go away. And he just dismissed this thought from the conversation. And that stuck with me, I think, because it really illustrates a shift, a sea change in our understanding of the world around us. I think that when I grew up, our view was that the world was vast. It was almost infinite. The sphere on which we live was just so large that our impacts could hardly matter. It wouldn't matter if we tossed out garbage, or it wouldn't matter if we cut down a particular tree, because there was just so many. The world was so vast. I think now it is very clear the world is finite, and this perspective has changed. And I think children today are leading the charge and really pushing us adults to recognize this sea change. So I am going to talk to you a bit about my work as an evolutionary biologist and a bit about how I see human impacts on the evolution of the tree of life. So we'll go together on this conversation this evening. So I am very fortunate to get to study the evolutionary changes that have occurred over the 4 billion years of this planet and have led to the vast and wondrous diversity of organisms that we see around us. I do so, might be surprising, but I do so using mathematical models largely. And the mathematical models are important because evolution is actually pretty complex. You know, we have all of these mutations at many loci, there's recombination, genomes are complex, the environment is complex, lots of species. And so it can be hard to predict when evolution goes left and when it goes right, metaphorically, when it leads down some paths and not others. And so math models are helpful in determining the paths that evolution can take and the paths that evolution cannot take to help us understand how this diversity originates and is maintained. We also use experiments. And since I started my career, it's just been amazing how we can peek into the genomes of the organisms that we study to find out exactly how they are changing. And I'll tell you a little bit about some work that we've been doing with yeast, the budding yeast shown here over in this little plaque. It's the yeast of wine and beer and bread fame. But it also is a really fantastic organism to study evolution in because it reproduces really quickly, about every 90 minutes. And that allows us to track evolution within a matter of days. And we use yeast then to test theory and expand our understanding of evolution. So the core of my research is really, finding out when evolution goes left or right on a number of different axes. For example, this axis, and this has to do with how organisms reproduce. You might think that evolution would have led to one common way of reproducing and everything would reproduce in the exact same way, but it's not true. There's enormous diversity in whether there's selfing or not selfing here, whether there's asexual or clonal reproduction or solely that or solely sexual reproduction. So why is there this diversity and what conditions favor predominantly asexual reproduction and what favor sexual reproduction? And similarly, let's look on another axis, genome size. There are many organisms that have one copy of every gene, which we call haploid. There's ourselves and many animals and plants that are diploid that carry 2 copies of every gene. But then there are some polyploids that carry many, even I think the record holder, a fern, that carries 1,200 copies of every gene of every chromosome. So this is remarkable diversity, and our models and our experiments are aimed to try and help us understand why these organisms with these different attributes coexist. And so I come to an understanding, an appreciation of the tree of life that's really awestruck, understanding the kind of different selective pressures that species have faced over their ancestry. And this really, one way to think about it is every single species alive today is a treasure trove of adaptations. They carry the genes that allowed their parents, their grandparents, their ancestors to survive. They've got the genetic solutions inside them to have survived through the environmental challenges, the diseases, the parasites of their past. And so this is a treasure trove that I see when I look at the tree of life. Just going to check the chat in case I am, good, okay. I can stop that. So this is just a little bit of background about where I'm starting from when I'm lecturing you today about adaptation, speciation, and extinction and how humans are shaping this evolutionary tree of life. As I mentioned, when I was a kid, I really, we were under the impression that the world was infinite. But now I think the news just reported this week that we've topped 8 billion people on the planet. And not only is the population size growing, but the ecological footprint per person, especially in countries like Canada, Europe, and others like that, that our footprint per person is growing. And that combination has meant that we have impacted the planet on a scale that we're only now beginning to appreciate. And just some quick statistics about this: 75% of ice-free land on the planet has measurable impacts from humans. 75%. 67% of the global biomass of the larger predatory fish has declined. 25% of the surface of the earth is considered degraded by erosion, pollution, compaction, or salinization. Basically, our human impacts have degraded the soil in these areas to the point where they no longer sustain the ecosystems that were there before. Temperatures have already increased by nearly 1 degree, projected to increase 2 to 5 degrees over the next century. And sea levels have risen by 0.2 meters and rising. Oceans have become 26% more acidic. And even in places where no human has ever set foot, these impacts are being felt. But what I would like to, before I go on, and what these impacts mean for the diversity of life with whom we share this planet are sobering. Here is a recent report from the World Wildlife Foundation and the Zoological Society of London. What they do is they collate all of the studies of populations that are collected over large periods of time to see how the numbers of a species are declining or rising over time. And this is summarizing over 5,000 species data sets showing that on average, since I was born, there's been a 69% reduction in the size of those vertebrate populations. So since I was born, many wildlife populations are just 30% of the size that they were once. You've probably also seen a similar report about the lost 3 billion birds gone over that time period. And if you are like me, you might remember just how many barn swallows were in the sky, or not birds, but bats in the sky, and how it's more striking when we see them these days because so many have been lost. How do we know this? We know this from a combination of citizen science, people like you who do bird counts, breeding bird surveys, or many of you have probably contributed to iNaturalist, those sorts of data. So that's part of why we know about these trends. And another is really remarkable, using big data science, using weather maps, satellite imagery that can literally see this light area are birds in flight. So from radar, we're learning about bird flight and how the size of those bird migrations are changing over time. So let's spend this hour thinking together about how humans are reshaping the tree of life. We'll talk about how humans are changing selection pressures, how humans are altering the extinction landscape, and what little we know about how humans are altering the speciation process. And maybe if you can only take home one image from this, it's that humans are reshaping this tree of life. Much like this espalier, this tree that we have that has been shaped for agricultural purposes. We're shaping the tree of life so that the branches aren't wild, higgledy-piggledy, but it's the branches that are surviving are the ones that survive best for us and with us. So this espalier is basically the poster child, unfortunately, for this talk. I argue that humans have altered the course of evolution to a degree that no other single species has ever done in the history of this planet in a single century. And that we are favoring the organisms that best tolerate our land development, fragmentation, harvesting, species introductions, movements, environmental changes that we cause. So these are multifarious, and I really want to emphasize that the ways in which we're changing this tree of life, that we're selecting on species is really so multidimensional. We're fundamentally changing who can survive and thrive and reproduce in the world around us. So I'll just go through some examples of how humans are changing. We call it the fitness landscape in evolutionary biology, but it's basically the types that have high fitness and survival and those that have low. Collisions, let's start here. Our built environment and our cars is known to cause the death of over 800 million birds in the US and 60 million birds in Canada annually. And it's not random which birds are killed. Those birds that are migratory, that spend a lot of time in air and that are less maneuverable are the ones that are more likely to be killed. And so studies have found that species are becoming increasingly sedentary, meaning they don't migrate long distances, for example, in these great bustards. And that collisions are a major source of mortality among those birds that do move. Other studies are finding that collisions are actually reshaping the body of birds. Cliff swallows have evolved smaller wings, shorter wings, in areas with high traffic, within areas of high collision rates, and also comparing survivors and the road mortality, individuals that are killed by cars tend to have longer wings, less maneuverable. So birds are evolving, are facing different selection pressures to avoid our built environment. The other way in which our built environment changes the selection pressures of organisms is actually to create new habitats that never did exist before. And my favorite example of this are mosquitoes that have colonized subways and underground environments and find a wonderful food source. As we wait for our subways, these mosquitoes are able to eat us. And they've now evolved this kind of subterranean lifestyle and changed their biting habits to evolve a preference for biting us. So that's the built environment. How else are we changing the selective pressures? This one is maybe a little more familiar to you, and that is that our hunting and harvesting has selected for species that stay out of our crosshairs, for species that avoid being captured. And that one of the earliest examples of evolution by kind of human Anthropocene selection was a local extinction of silvercoat variants in foxes, because the silvercoat variant was so highly prized by those seeking fur coats. I was just in Uganda and remarked about how many of the elephants that I saw had no tusks. We have been selecting for shorter tusks and no tusks in elephants because of the harms that come from bearing tusks and being susceptible to poaching and hunting. So shorter tusks have evolved, and studies have found that there's been decreases by about 25% over a generation. That's incredibly strong selection on elephants for, or I shouldn't say for, against their tusks. Some lighter examples, fishing. Fishing selects for less catchable fish. Fish in general are evolving to be smaller, especially when harvesting uses nets with a certain size that's selecting out the large fish and allowing the small fish through. It's also selecting for smaller mouthed fish to avoid the hooks. For example, a study of bass showed that the smaller mouth can help the fish survive and avoid our fish hooks, as well as behavioral changes, faster and bolder rainbow trout. So these are just a sampling of the literature that my colleagues have amassed of just how we're changing the selective landscape of the world around us. We're also creating novel communities, moving species around in new ways, and this is also altering that fitness landscape. And this is well documented in many different communities because of the introduced species and altered ecosystems that humans are causing. But just as one example that I'll highlight here, cardinals. These beautiful red cardinals, they're not here, but I think they are in your neighborhood. The best conditioned males have been taking up territories and introduced honeysuckle. But honeysuckle is a very bad bush to choose because predators can more easily catch and kill these cardinals. So a study found that there was actually selection against the highly colored, red colored, most fit males, because they were establishing their territories in these honeysuckle invasive plants that was allowing them to be killed by predators. So we're reversing this selection pressure for bright red coloration in cardinals in this study population. I'm looking out my window right now, and most of it is concrete, and there are little patches of plants and trees in my neighborhood. And that is actually shaping selection and dispersal of plants in urban environments. For example, in this Steacie, the seeds that are being produced are more likely to land straight down and not fly off in the wind, because the existence of that plant to seed means that it's growing on land and not on asphalt. And so we're seeing the evolution of dispersal ability, especially in urban environments where there's less dispersive seeds. Migration timing has been affected by climate change that humans are caused. There's been a meta-analysis showing across studies that migration timing has shifted about 2.3 days per decade. So that's when the species and move. And as one example, pink salmon here on this coast migrate 2 weeks earlier than they did 40 years ago. And the genes underlying, most of the time, a lot of these changes, we don't necessarily know if these are genetic changes or behavioral learned changes. But in the case of the pink salmon migrating, the genes have been identified that promote that earlier migration. So it's a genetic evolved change. We're also shifting inheritance. As I mentioned, I'm interested in genomes and how they evolve, and many of, especially our crops, are larger in genome size. They tend to be polyploid, and we're even shaping the amount of recombination in the species around us, how much genetic mixing there is. So we're broad scale finding that when humans change the world around us, that who survives and who dies in those species are shifted. And that can be very strong, very rapid selection facing the planet. And of course, not all species are responding fast enough. And some of the work that my colleagues and my group are interested in knowing is what is going to determine who are going to be the survivors and who are the losers in this reshaped world. There's this metaphor that we have that if a population is failing to respond to selection, it's not keeping up, that it will start to decline in size as seen by these dots, and that can lead to extinction. But it's possible for evolution to rescue populations if they evolve fast enough. Perhaps those pink salmon as an example that adapted fast enough to migrate earlier. And so we're interested in those cases. When can evolution favor and promote adaptation? And one of the things that this is required is kind of a wholesale new set of mathematical models that account for these population size changes, as well as look at how selection and mutation are shaping evolutionary change. And so we're adding to our body, to our understanding of evolution by incorporating these changes that many species are facing. And trying to identify when evolutionary rescue can indeed occur, where those mutations are ample enough and soon enough that evolutionary rescue happens, reversing these population declines. So just a short little break to talk about some yeast work in my lab. This is work that my now colleagues, former students, Lisa Gerstein and Jasmine Ono did while they were in my lab. And they were working with yeast that whose environment they would change. And they would ask under what circumstances yeast could adapt to those changes or when they would go extinct. They used a fungicide, nystatin, and a heavy metal, copper, which is very commonly found in mine tailings or in other polluted sites. So we put yeast, I have to tell you that these are wells. They are wells that we make identical. They have the same environment, the same amount of copper, and we put a bit of yeast in each well. And we come back a few days later and look. And just like at the bottom of a beer bottle, if you have yeast in it, the yeast will settle to the bottom. And that's what we're looking for, because if they do, that means they adapted. We're able to grow, proliferate, and make enough yeast that we could see them. So you can see some of these populations were rescued, the ones that with white at the bottom, whereas many of the other wells were not. And again, these days, we can sequence each of these lines and figure out who got rescued and how did they get rescued, looking for what characterizes those lines, those yeasts that are able to adapt in this rapidly changing world we put them in. And one of the questions that we're asking is, when we are changing the environment, not just in one way, but in multiple ways, are organisms able to keep up? Are there combinations of environmental pressures where there are really no genes available, no genetic changes available that allow adaptation, that enable adaptation? So, for example, in the experiment that I talked about with copper and nystatin, if we adapt to nystatin, are there any mutations that are helpful for copper? And the answer was yes, there are mutations that will improve fitness in both copper and nystatin. But when we look at another environmental combination, high salt environments, as well as this fungicide, we actually didn't find any mutations. Nowhere in the genome was evolution able to rescue those populations. And so one of the things we're trying to figure out in the lab, just as a start, is what is accessible to evolution and what is not, to better understand the combinations of environmental changes that really will pose the biggest challenges. We're also able to ask a different question in the lab, which is, are there some attributes of an organism that make them easier or harder, less able or more able to adapt to these changed environments? And Jasmine, in one of her experiments, showed that the diploids, ones with 2 copies of every gene, were actually not able to adapt ever to nystatin. We didn't see any evolutionary rescue in the diploids, only in the haploids. And that makes sense from some theoretical point of view, because the haploids expose every single mutation that appears. They only have 1 copy. And so that beneficial mutation really helps them. But this indicates that there are going to be species that are more readily able to adapt to our changed environments, and others that will not. So as we alter the world and some species go extinct, it's not random extinction that is going to happen. We know that from studies across many taxa it has been estimated that 1 million species are now threatened with extinction. This is really unprecedented extinction rates, much higher than background extinction rates. And we are called together to really make the transformative changes that we need to do to restore and protect nature. I know that the COP meetings soon to come to Montreal will be grappling with this very issue. So we know that extinction is not random. It's hitting some groups more than others. And this is a compilation of the different groups. The redder and orange colors are the more extreme extinction risks faced by different groups of organisms. Amphibians and cycads down here are really being hard hit. But across the board, over 1 in 5 species of vertebrates, invertebrates, and plants are at risk of extinction, including vulnerable, endangered, critically endangered. Here is a map in Canada of where those species live. They live mainly along our southern border. That's where the highest density of species at risk are, basically where we are. Just to bring faces to these numbers, little brown bats have declined 94%, largely due to white nose syndrome, an invasive species that was introduced by humans into North America from Europe. Swallows, I've already mentioned, many swallows have declined by over 90% in our lifetimes. Eulachon in the Fraser near where I live, endangered with 98% declines in the last 10 to 20 years. And of course, orcas that are endangered and down to less than 100 individuals. Lotus pinnatus, an endangered species in the Garryoke ecosystem around in the Gulf Islands near here, and bear's hairstreak, another endangered species from here in the Okanagan. And I illustrate and show you these because endangered species aren't a world away. They're not just in the Amazon where deforestation happens. They're here in Canada, and we need to work to save them here. Just quickly, what are endangering species? There are a lot of threats to species, and they're listed here, both in Canada and across the world. Many of those are exploitation, harvesting at too high a rate, eliminating habitat is in green. So direct effects like those. Climate change in purple, here this little one, is a small factor so far in driving these species to be at risk. But we expect this to be an increasing pressure on the species that as the climates warm, the oceans become more acidic, because many of those changes are ramping up, and we're just starting to see a larger and larger mismatch between where species are and where their optimal environment is. So in in British Columbia, for example, studies of the speed of climate change have found that the optimal climate for a given tree species is moving north at 1 kilometer per year. Well, trees don't move that fast, and seeing an increasing mismatch between where trees grow best and where that best environment is for them, 1 kilometer a year. So this leads us to expect extinction risks to accelerate over the next century. And Mark Urban has estimated that in addition to those threats that we've also already talked about 1 in 6 species will face will be at threat simply because of climate change. So I've mentioned that this extinction risk is not random. I've mentioned groups like amphibians and cycads that are being heavily hit. But it's also not random in terms of the attributes of the species. that are more often going extinct. The species that have already gone extinct from overhunting are the large-bodied organisms: sea mink, passenger pigeon, great auk, dodo bird, no longer something that our children can see on this planet. So large body size. The largest declines in terrestrial species population size have been seen in those species with large body sizes. And that's true over millennia, and it is also true today. In addition, species with small ranges, like the species that I mentioned earlier, the bear streak, and other species with narrow ranges, like these Gulf Island endemics. Woody plants, oddly, woody plants are more likely to go extinct. They are more often on the endangered species list than you'd expect. Tropical plants, dioecious plants. Dioecious plants have separate male and female plants, like willow trees, for example. And it might just be harder for them to find each other in the more fragmented landscapes that humans are creating. So we're not randomly taking or pruning off branches of this tree of life, but we're pruning in very unusual ways. Widespread, who's going to survive? Widespread, generalist, dispersive species that get along well with humans, that are human commensals, are on average less prone to human-caused extinction. So that's the extinction side of the picture. And we've talked about selection. We've talked about non-random extinction. Well, what's about the generative side of evolution? What about the speciation process? What are humans doing to the speciation process? Because we know that species are always being formed, and that balances to some extent the extinctions that are happening. So are speciation rates going up dramatically? Let me tell you what little we know about this. We do know of some new species that are formed. Many of these are formed because of the new niches, the new environments that are created by humans. For example, apple orchards have been infested with Ragelitis, a new species, Pomonella, that prefers apples. So we selected for apple trees, and that generated a new food source for Ragelitis. Cereals, the fungi that infect cereals have diversified as we select for different types of cereals, different types of wheats, etc. And the fungi have speciated alongside our selection for different cereal crops. Mine tailings, these really, really polluted areas, have also been colonized sometimes by these remarkable organisms like this grass that are able to tolerate and evolve characteristics that allow them to survive and thrive in these new environments. So we have selected for new species. But we've also done the opposite. We've collapsed niches. And this is often done completely naively, where the signals that species relied upon to keep them apart disappeared. The most famous example of this is in Lake Victoria, where eutrophication, the kind of runoff from agriculture and urban waste, that pulse of nutrients led to murkier, cloudier lake environment. And that led to the collapse of likely hundreds of species of cichlids that could no longer tell each other apart. And those really flashy cichlid fish traits that are sexually selected and were used as cues to prefer to mate with one's own species have gone away, and this has led to a major collapse of those species. Here in British Columbia, we had a similar story where we have these lakes where stickleback species have diversified, and we have ones that live in the benthos near the plants on the edge of the lake, and ones that swim in the middle of the lake, limnetics. Well, in Enos Lake, a The spread of crayfish led to the decimation of the benthos environment, and the two species collapsed together into a hybrid swarm. So we're reducing species. Fragmentation is another way in which humans are influencing speciation. By this, I mean that populations that were before one and the same and contiguous are, through fragmentation, no longer exchanging individuals, no longer having gene flow, and that is allowing them to diversify and differentiate. We're seeing this across the Panama Canal, for example, with Jeffrey's tamarin, where there's evidence of more differentiation on either side of the canal. And we're seeing this in sticklebacks on either side of dams, for example. But that is also flipped around. The fragmentation that we're causing in some habitats may be increasing speciation, but on the other hand, species that were isolated on different continents are now being brought back together, again, leading to a lot of hybridization and collapse of species. And Spartina is a very good example of that. So actually, we don't know. We don't know whether or not humans in general are increasing the rate of speciation or decreasing the rate of speciation, although we have examples of both. In the lab, we are studying what strong selection can do and whether or not that tends to promote species or not. How we do this is select on lines kind of like north of the Panama Canal and south of it, where we keep lines separated and evolve them for a period of time. And we're trying to figure out how selection acts on these lines and whether or not they, when they hybridize, are incapable of producing hybrid offspring. And that is a sign, a characteristic signature of speciation. If they evolve and through that evolution are less able to produce viable offspring, then that's the hallmarks of speciation. And so in the lab, my students have done this. We've done this with nystatin again and looked at different genetic routes that different lines took towards nystatin. And I won't, I just want to orient you briefly to what I'm going to show you in this one result slide. The ancestor that can't survive nystatin. It's always at the bottom of these diagrams. One line that had a mutation in one gene is shown on the left, another line that had a mutation in another gene is shown on the right, and then we cross them and make them their offspring, some of which have the double mutant. So we're interested in, are those hybrid double mutants more fit or less fit? And across the board, what we find is that those double mutants are actually less fit than we'd expect. If it were independent, we would expect a nice parallelogram, and none of these are parallelograms. Those double mutants are always a little less fit. So the hybrids between these lines that we've evolved are a little less fit, and sometimes, even though they carry mutations that each of which helps them in this new environment, together, they're actually less fit than either parental line. And those types of interactions, we call them BDMs for short, doesn't matter, but Bates and Dobzhansky-Muller interactions. But these interactions where you adapt, where lines adapt, but when they come together, their hybrids are sick, I would argue are going to be really, really common when selection is strong, like it is in our experiments, but also like it is in many anthropogenically altered environments. And that is because when you put two lines that have adapted through very, very rapid evolutionary change, they have large effect mutations. These large effect mutations, when you put them together, are more likely to overshoot that environmental optimum and decrease fitness and reduce the fitness of hybrids. And we're seeing that in the lab. And we're also seeing it in simulation results. That I'll just show here. The faster the environment moves, the faster we're seeing these kind of hybrid breakdown, or these first baby steps to speciation evolve. And so theory predicts that indeed strong selection and evolutionary rescue are more likely to lead to new species. But I should say that while we don't know, what we know examples where new species are formed at a higher rate because of recent human activities, and where many species have collapsed, like the Lake Victoria cichlids due to human activities. I've mentioned that we don't know much about the net effect, but that strong selection has theoretical reasons to lead more often to very differentiated populations whose hybrids break down. But what we do know, while we know, very little about the net effect on speciation, what I do know is that every new species that is formed is only a twig on this tree of life. Every new species that humans do facilitate in creating has very little evolutionary uniqueness, very little evolutionary time to differentiate from its sister species. So, whereas when a species goes extinct, it is taking with it millions of years of adaptation that make that species unique and differentiated from any other. So speciation isn't going to balance out the extinction story, because when extinction happens, it takes away that treasure trove of past adaptations. And when new species are born, they're babies with very few differences. So just to wrap up, we have altered the course of evolution. We have reshaped the tree of life by reshaping the selective pressures that species face, the extinction risks that are posed differentially, more heavily on some species than others, and even changing the nature of speciation. And this legacy of an altered evolutionary process is a legacy that we are leaving to future generations in a world that is less natural and less wild than the world in which we were born. And I think that together we have to work as a community to place limits on our activities, on our impacts on nature, so that we can make room for wildlife and the species with which we share a planet. So I haven't gone through all of these final points, but I think I will leave it there and open it up to questions. Thank you.
Thank you very much, Dr. Otto, for such a engaging and dynamic seminar. It was excellent. I've been monitoring the question and answer section, so we have a few questions now, and everybody who's watching, if you have additional questions, please put them in the Q&A section so that we can get them to Dr. Otto to answer. Do you want me to read them?
No, I'm just reading Tess's.
Okay.
I hope I can mention your name. The first question: has lack of diversity in monoculture agriculture affected selection in wild species, such as species interactions with fields of monocrops or flowering plants interacting with an abundance of honeybees instead of native pollinators? And the reason why I'm like, wow, good question, is that one of the postdocs in my group that I have been working with, Julia Kreiner, is about to publish a paper in Science. It's going to come out December 8th, where she has shown that a wild species, water hemp, has its evolution has been shaped by agriculture, and so it's more likely to be herbicide resistant. It's also more likely to be drought tolerant. Water hemp, it's a plant that lives normally in riparian habitats, but because agriculture is such a predominant landscape feature now, this plant has shifted towards living on farms. It's considered a weed, it's considered noxious, but it has also evolved to be drought tolerant because it's no longer in those riparian areas, but living on farms. It also is growing faster as a little baby juvenile plant in order to outcompete or to grow with alongside those crops. And so, yes, I think your question is right, spot on. agriculture, we think of domestication and the processes, the ways we've shaped domesticated crops, plants, and animals, but actually our agriculture is shaping the selection pressures that what we would consider wild plants are facing, are also experiencing. And you raise good points as well on honeybees and kind of shifts in which pollinator preferences. I don't have anything about that particular story, but you're exactly right. You're thinking along a good research line of investigation. How has the switch to, again, honeybees are non-native pollinators in North America, how has the abundance of this new non-native source of pollinator altered selection pressures outside of agricultural fields? Do you, sorry, I haven't monitored. Do you want to get highlight the next one?
Eduardo is next. And can you go back to the part about likelihood that species develop mutations that are beneficial versus not beneficial, and the idea that an evolutionary path can be rescued? Can you repeat the main takeaway from that part, please?
Yeah. So, so if most of the evolutionary theory was developed assuming populations were healthy and assuming that they were all of a large and kind of constant size. But remember that Living Planet Index that said that we'd lost 16, that vertebrate populations on average have declined in population size by 69%. Well, that's the kind of thing that I'm talking about. As those populations decline, you project that forward, and that is leading towards extinction. So we need models and predictions about which species are going to be able to survive. Now, some of that decline is due to habitat reductions, and there may not be an ability for that species to adapt and change. But if it is able to migrate at a more suitable time, if it is able to avoid collisions with our buildings, if it is able to avoid our hooks by evolving by genetic changes that increase survival, then those are what we're calling evolution. That's what the field is now calling evolutionary rescue. Evolution is sometimes able to rescue these populations from decline by mutations arising within those populations that before they have gone extinct, that allows them to survive and thrive in our world at present. So that's what evolutionary rescue is talking about. As populations are facing an increasingly inhospitable environment, will or will not evolution allow them to keep up? This is pretty dark. Yeah. It is pretty, yeah, it is pretty dark. And maybe I think this final slide is worth emphasizing. We can act together. First of all, the number one thing we can do is raise our voices, raise how important it is to keep natural spaces available and free for wildlife. Reducing our footprint as much as we can, working together. I work with the Nature Trust of British Columbia to try and protect some of these landscapes where there's a lot of endangered species, to give them a place to live and survive and make it into the next century. So let's see, would you venture some actionable suggestions about what we can do to create a bit? I think also I do think that part of this is a problem of my generation growing up and thinking the earth was infinite, and it's not. And I am hopeful that the university students that I talk with are very active and engaged, and I think they know we are living in this one planet is precious, and that we must protect. And while it is dark, I also want you to move away. Evolution is this amazing process that over 4 billion years has led to this treasure trove. Share with me that love of that treasure trove. Share with each other that love and appreciation of every little species that shares our planet, because we need to fight for them. We need to be their voice.
We have Andrew. I'm not sure if you're reading that one or if you'd like me to.
Why don't you read that one? And
Okay.
I'll just take a second
Thanks
break.
for your interesting and important talk. You mentioned that there may be winners and losers in terms of adaptation to anthropogenic change. It would seem that anthropogenic selection compounds the selection already generated by organisms' natural environment, suggesting that this adaptive response provides an advantage only in relative sense and comes at a cost in terms of their fitness in an absolute sense.
That's right.
So would you say that, you go
Yeah.
ahead.
You know, I think we need to ponder this. We need to consider it. Yes, we are changing that selective landscape, and the birds that are surviving car collisions have shorter wings. But what does that mean for them? Are they less able to stay in flight for long migrations? Are they less able to gather the food that they need, or more able? So how does that change their ability to move across the landscape, given that they're naturally selected optimal was for longer wings? So what are the trade-offs that are kind of imposed on species when we shift the selective surface? Those bass that are evolving a smaller mouth, what are the costs for them, and how is that changing what they can eat and who they are eating? So yes, I think I don't think we have, as a field, I don't think we've grappled with this yet. But you can think of it as an anthropogenic fitness cost. We're either intentionally or non-intentionally changing what can survive and what can reproduce, and that is moving species away from what was naturally selected and naturally allowed reproduction. Oh, I see one that I want to go to, and that is, there was one about citizen science. And I just have to say, if you have a phone, I love this app. If you don't have it, please get Seek. Seek is connected to iNaturalist, and you can load all of your findings on, I'm just going to pull it up, all your findings on, I don't know if you all can see that, onto iNaturalist. But the coolest thing is you just turn on the little camera button, and if you are outside, you point it at the plant that you don't know, or at the bird. Well, the bird's not going to stay put, but if you are lucky enough to capture the bird or a bumblebee, this will give you a species ID and allow you to upload your photograph. And it is then verified by experts that can use the GPS location of that photograph and your photograph. So, you know, my mom sends me her pictures every day for the things that she's like, what's this? And Seek says this. And so anyway, it's something everybody can do. You don't have to be a biologist in order to use these programs and to contribute to citizen science. There's another one too, Merlin, that lets you listen in on bird song, and it translates those bird songs into the species singing. So really, we can all participate.
Fantastic. Christine is asking, thank you, that was wonderful. How well do you think the yeast data, back with such short generation times, predict the fitness effects of isolation in longer lived wildlife species?
Yeah, so we've kind of got this mismatch between the types of organisms we can really quickly do experiments on to figure out what will go extinct and what not, and the large-bodied species that are facing the biggest extinction risk at the moment. Well, I should say, actually, we don't know much about the extinction risks faced by fungi, because we typically don't look in the soils beneath us that carefully, and we move soil around. So maybe the fungi are facing a higher extinction risk than we even are aware of. Anyway, there is this big point. So I don't see those experiments as necessarily telling us everything, but chipping in a piece of evidence. I think things like it will be harder for diploids to adapt to a fast changing environment is probably generally true. It matches the theory, and the experiment, it was really clear. Like, we repeated the experiment and repeated the experiment, increased the population size, and we have now done so many replicates that we are sure we've hit every single base pair in that genome. And yet the diploids never were rescued in that particular environment. And then I think the other thing that we collectively as a field need to figure out more is which combinations of environments are really dead ends, are really almost impossible for organisms. And is there rhyme or reason to those? Can we, do we have kind of a foundation to predict which environments will be particularly challenging, which combinations of environments will be particularly challenging? So I think, we already know for a lot of species what we can, the large bodied species, what we can do, and a lot of it is habitat preservation, rehabilitation, and expansion corridors. And so it's not an evolutionary question, but really an ecological one. But I'm speaking here about the evolutionary aspects.
I guess there was a latter question to that, and asking what you thought the sort of timelines would be in longer lived populations, century or two, or would it take longer than that to see?
Yeah, that's a really good question, because we're sometimes seeing this in yeast within a day. So we're not talking, it's in these evolutionary rescue experiments, they're so extreme that only the cells that soon carry a mutation, or that already do carry a mutation that allow them to survive, are making it. So I think that this is another aspect of evolution that is increasingly appreciated. It is fast when the selection pressures are strong. And so it really, like with white nose syndrome, which is Christina's expertise, not mine, really, you can be talking about changes that are occurring within a cave, within a season, among the individuals that are surviving or are not surviving. So I think it used to be considered that evolutionary change was, something over a geological timescale, and that's not true. It's happening here. It's happening now.
Great. Lauren is asking, hi, Sally, thanks much for the excellent talk. I'm wondering, in your experience, what the biggest public misconception related to speciation and or evolution? What's the best way to communicate with others to dispel this?
Well, first of all, I think it's gotten better. When I was a graduate student, I actually wouldn't introduce myself as an evolutionary biologist. I would introduce myself as a population geneticist, because there was such evolution was really this, considered to be this, I don't know, anathema, or you just would have it politically charged. Let's put it that way. I don't think it's politically charged anymore. I certainly don't think it's politically charged in Canada. We've seen so much evolution in the news with SARS-CoV-2. We're just not in the same place we were in the 1980s about understanding evolution. The biggest misconception is it's something that happens over so slowly we don't have to think about it, and that's not true. We can study it, as I mentioned, we can study it over the course of days. Those were baby steps in the lab that I was talking about towards speciation, but we can study speciation in the lab again over the course of weeks. So I think that there may still be some circles that doubt evolution as a process, but I think it's we just need to speak more about what we as evolutionary biologists do and what we know. So anyway, I guess I would say I think we've come a long ways, okay, but we still have a ways to go.
Maristella asks, are there noticeable evolutionary changes in human population which are a consequence of human behavior in the past century?
No, I'm alive. I'm alive, and I wouldn't be. And that's because when I gave birth to my son, there was a mismatch between head width and my hip width. And I had a C-section, survived, no problem. But we do know that that human activities and medicine are helping some of us survive that otherwise wouldn't. And that is leading to shifts in some traits in the human population, but I don't think so. Yes, humans are evolving, and those of us who survive and reproduce are different than who would have before. But medical practices like that, I think I'm glad I'm alive. So that's not that altered evolution is not necessarily a bad thing. I'll say that. But the other thing, the main thing to know about humans is I've been talking mostly about genetic evolution, but cultural evolution is much faster. And it is also what makes me hopeful, because if we have a mutation of an idea, if we can let it spread and help it spread, and that helps people contribute more to protecting the environment around them, that's the type of change that we need to foster, cultural evolution that helps us protect the species around us.
Excellent. How are vaccines affecting the evolution of COVID and other viruses, germs? That was Michael's question.
Yeah, so I would say we know that, I'm not going to say vaccines, but immunity, and that's from a combination of vaccines and the very high level of infections that we've had in Canada. So there's been about 70% of the Canadian population has now been infected by SARS-CoV-2. But if you count up everybody who's been exposed to this virus, either by infection or vaccination, it's really close to 100%. So in such a high immunity background, we have changed the selection pressure of SARS-CoV-2. And what we're seeing now with Omicron, we haven't seen before, and that is that what variants are spreading are the variants that are different in this one place where our antibodies attach to the outside of the spike, the receptor binding domain, and our antibodies clear the virus and prevent it from entering our cells. So I don't know if you've heard, but there's been a lot of convergent evolution, BA2, BA4, BA5, all evolving the very same changes in the receptor binding domain. And that convergent evolution of the very, very same mutations is happening because immunity is so high at the moment. So yes, we are also shaping the evolution of SARS-CoV-2.
Rosalind is asking, she also says, thank you, Sally, for this wonderful talk. It was fascinating to see the tree at the end that highlights how twig-like current speciation is. What does it take for a new major branch to form in the tree of life? Mass extinction plus a lot of time? Question. Sorry if this is also dark. It was really eye-opening to see that metaphor visualized. Yeah.
So there is nothing but time that will lead to a long branch, but so we have to protect those unique species, those the treasure trove around us, because we can't get it back. If they go extinct the millions of years, we'd have to wait millions of years for those adaptations to accumulate again. But I think you are raising these points about, in the geological timescale, what have been the major drivers of new species formation. And yes, a lot of times they are pulses of species formation, for example, after mass extinction events, where we think that major new niches are open. And so there would have been baby species if we go back right to the KT boundary. There would have been baby little species at that point too. But they've had now the chance, the millions and millions, hundreds of millions of years to accumulate mutations and diversity.
Rachel asks, can you give some examples from your research or others where evolutionary theory can be harnessed to facilitate solutions to the biodiversity crisis?
Yeah, Rachel asked a really, really good question, because I would say for many species that are, I mentioned habitat and over harvesting were the '1 risks facing species. If that is true, evolution's not going to save them unless they evolve to avoid our hooks and our firearms. But by and large, it's a question we know the answer to. We have to stop the harvesting. We have to stop the habitat destruction. So that's an ecological, environmental conservation action that we need. So what evolution can do is, there are questions about helping species that are not as migratory to shift as their ranges shift. So I mentioned in BC that the climate was warming so much that the optimal environment for species was moving 1 kilometer north a year. So forestry practices in this province have now shifted because the evolutionary and genomic advice was to not source from seeds from that area that's around the trees, but to source from further south to make them better adapted to a warmer climate. So we've shifted, and now I think half of the seeds are locally sourced and half come from a further south environment. So that's called assisted gene flow. It's not moving species outside of their range, but it's helping them get maybe more genetic variation that's valuable to them. And I think for long-lived species like trees, species that don't migrate very much, we may need to give them a helping hand.
Okay, and we have a question from Tess. So her question is, are you aware of any studies, research being done into the loss of species used in Indigenous cultures and medicine? How much biowealth are we losing in Canada by allowing these extinctions to continue?
Yeah. Yeah, I think that we only know the tip of the iceberg. We only know, it's been estimated that we may only know about 10 to 30% of species on this planet. So we're losing things and we haven't even registered. So I think what you are raising is this important point too, that the adaptations that allowed them to survive to past predation events, past diseases, some of those adaptations are useful for us too. They are what our antibiotics come from. They are where our healing medicines come from. And so yes, an excellent point. Those evolutionary treasure troves are also useful for us oftentimes, but we may not know it. And again, there's two aspects of what we're losing. Not only are we losing species through this biodiversity crisis, but we're also losing that knowledge because of the many ways in which Indigenous communities have been subjugated, and their culture and their connections to that knowledge broken. So we need to do everything that we can to support the connection of those communities while it still exists.
Wonderful. Thanks, Bruce, for moderating that fascinating Q&A. And of course, Dr. Otto, thank you so much for giving us some really fantastic insight into the complexity of evolution, and probably more importantly, for challenging us to commit or recommit potentially to action.
Yeah. Thank you.
Needed to protect our natural world. Thank you so much. It would have been great to host you in Ottawa, but we're so glad we could have you nevertheless join us online.
Oh, my pleasure to be here.
Yes.
Thank you so much for the invitation.
Thank you so much for your time. And to our attendees, I hope you enjoyed this lecture. We really appreciate you all joining us. And if you're interested in more events like this, please visit our website at carleton.ca/science or follow us on social media at Carleton Science. Thanks, everybody. Have a great evening. Enjoy nature. Thank you. Thank you so much, Dr. Otto.